So I'm very, thank you very much, Johan, for that very gracious introduction. I'm very honoured and mildly embarrassed to return here to the Karolinska to give this lecture. The last time I was here, I think, was in 2012, with exactly the same title of today's lecture, which I discovered subsequently. But I can tell you there's only about 8 slides which are in common with the 2 lectures. And I'll give a special prize to those of you who remember them all. So impulsivity and compulsivity. The reason I'm interested in these is because a bit of a revolution in psychiatric diagnosis may be going on against the older concepts of categorical diagnosis, and thinking more about dimensions of symptoms which generalize across different categorical diagnoses. And one place to start there may be with some of these traits, in particular impulsivity, which, as you can see from the Web of Science citation, is a very highly cited construct, impulsivity. It's generated a great deal of research interest. And in my last lecture in 2012, I focused on this construct. This time, however, I'm going to focus on a newer dimension, which I'm going to call compulsivity, which, as you can see, has far fewer Web of Science citations. And I need initially to distinguish these 2 constructs in terms of what they mean functionally, and then later in the brain. So impulsivity, a working definition, actions which are poorly conceived, prematurely expressed. In other words, actions which occur before they should. Unduly risky, often inappropriate to the situation and result in undesirable consequences. That's not to say impulsivity is always bad, but sometimes it is. It can lead to disastrous decisions. And the prototypical disorders for impulsivity, of course, include attention deficit hyperactivity disorder, mania, personality disorders, the Parkinson's dysregulation syndrome, and substance abuse, as we'll see. And the various psychological concomitants of impulsivity I've listed here. I'm not going to describe them in great detail in this lecture, but they include the discounting of reward, the control over actions, timing impairments, and so forth. Now, impulsivity shares with compulsivity the need for impulse control or behavioural inhibition, top-down control, probably from prefrontal cortex. But by contrast with impulsivity, these are actions which persist inappropriate to the situation, have no obvious relationship to the goal, and often result in undesirable consequences. So this is the distinction. And I think psychologically we're talking about behaviours, on the one hand, with impulsivity, which occur before reward, before an action is initiated, controlling those, whereas with compulsive behaviour, we're describing actions which are occurring at the end of the sequence, after feedback, and therefore what happens next. Do we continue with the behaviour or do we switch to something else? Now, the prototypical disorder for compulsivity is OCD, obsessive compulsive disorder, and much of my lecture will be focused on this disorder. But you can also see evidence of compulsive behaviour in frontal lobe dementia, schizophrenia, autism, and eating disorders, gambling, and certainly substance abuse. The new diagnosis of substance use disorders in the DSM-5 has about 13 symptoms, and I would say about 7 or 8 of them can be linked to control of compulsive behaviours. This is a major definition for substance use disorder. So these dimensions are almost certainly controlled in part by frontal lobe structures. On the right we have the human reward system, defined in terms, for example, of fMRI studies, with the head ganglion in the orbitofrontal cortex, and these all-important links in the ventral stratum with limbic inputs in this hot loop structure here, modulated by the monoamines, in particular dopamine, and paralleled by other cortico-striatal loops which mediate motor function, including the putamen here, and also complex aspects of cognition, let's say, also involving different sectors of the frontal lobe. Now, my basic premise in this talk is that impulsivity and compulsivity are governed by different cortico-striatal loops, different systems in the brain, and they're under different chemical modulation. And to illustrate this hypothesis, because it's just that, I've turned the loops on the side here, and you see impulsivity at the top and compulsivity at the bottom. So the argument is that impulsivity, which by the way is not a unitary construct and probably consists of a number of different systems and deficits, these are mediated, we argue, by ventral striatal structures, and perhaps including some of the caudate nucleus, whereas compulsive behaviours relate in particular to other caudate structures, but also to the putamen and its role in motor loops. That's the basic hypothesis. And I can say that our prime theory of substance use disorder, or what I'm going to call addiction, if you forgive me that, is that addiction results in a devolution from impulsive behaviours, gradually with the chronicity of addiction to compulsive behaviour, by transactions between these loops. So, of course, a lot of these ideas have depended on animal work, and the discovery of the reward system is based very much on rodent work with brain stimulation reward, but also with drug self-administration. This is arguably one of the best animal models in the whole of neuropsychiatry, the intravenous self-administration model by which rats will voluntarily self-administer drugs for abuse. And this is very predictive of abuse potential, as we know. But Herbal discovered that rats would also self-administer drugs directly into the brain via implanted cannulae, notably amphetamine, in nanoliter amounts, but only to the ventral stratum. So this is the hot spot of the brain, which of course is in receipt of this important dopamine projection from the midbrain, from the ventral tegmental area. And as we know, other drugs of abuse may modulate this pathway through their receptor systems. And Everett and I were able to, with Gavin Phillips, to replicate some of this work and show that rats actually regulate the optimal level of dopamine in this structure by their behaviour. Now, we argue that this system in particular is involved in the anticipation of reward and therefore the regulation of impulsive behaviour about reward. And one way of studying that is in a situation where rats have to detect brief visual signals presented randomly in space in a situation where they're working for food, which is delivered in this receptacle here. And when the light occurs, the rat responds there and is given a food pellet. But what it must not do is respond either in adjacent holes or prematurely. If it responds prematurely, it's punished by being plunged into darkness for a few seconds before he can reinitiate the next trial. And the hooded rat, this is Zippy, one of our hooded rats, show a very interesting bimodal distribution whereby some of these rats are very impulsive. They show a good deal of this premature behaviour, which is maladaptive. And you have about 10% of them in this large population showing this persistent impulsive behaviour, which is maladaptive because they lose food pellets. And Jeff Daly and I and others have recently done a genome-wide scan on this behaviour using breeding of these animals. The trait breeds true, and the relative chromosome involved is chromosome 1, and we've identified a number of genes, including genes which modulate the GABA receptor, the mGlu-R5 receptor, and a number of synaptic proteins. But the really interesting thing about this phenotype is that it predicts the propensity to self-administer cocaine. So if you screen these rats and only then expose them to cocaine, they will take a lot of it. And in the original study, as you can see in the green dots, these are the high impulsive rats compared to normal. And this is also correlated with a reduction of striatal dopamine D2, D3 receptors in the ventral stratum of these rats, not in the dorsal stratum. And this is interesting because, as you know, Nor Volkow and others have shown similar changes in chronic drug abusers, but the problem with that work has always been to know, was that caused by drug exposure or was it pre-existing? And here's some evidence that it can be pre-existing, at least in the rodent. Those studies, of course, are difficult to do in humans. So we've translated this work to drug addiction in a number of ways, and I'm just going to give you a snapshot because I spoke about this 5 years ago. But this is work of Karolinska, who tried to find a way of studying this endophenotype, if you like, for impulsivity, for stimulant drug abuse, by studying stimulant drug abusers, but also their siblings who do not take drugs, so they're drug-free, as it were, as well as healthy controls. And we used an assay, if you like, of impulsivity called the stop task, the stop signal task, where you have to respond as quickly as possible on the left or right button as dictated by the computer, till you hear a beep which says, don't respond. And by modelling the response time distributions, you can determine not only a go reaction time, but a stop reaction time, how good you are at stopping a behaviour once it's been initiated. And as you can see, stimulant drug abusers are terrible at this. They have long stop signal reaction times, but so too do their siblings who don't take drugs, suggesting an endophenotype. And moreover, this is correlated with the loss of white matter as measured with fractional anisotropy in the arcuate sulcus, mainly projecting to the frontal lobes. And we know from a variety of other studies, including brain damaged patients, that damage in particular to the inferior frontal cortex does result in an impairment on this task, suggesting it's part of an inhibitory control system. So part of the idea, therefore, for stimulant drug abuse is that this propensity to make impulsive decisions, which may interact with the propensity to take drugs, may induce people to take drugs, and may also lead to relapse. That's the hypothesis. But what I'm focused on today is compulsivity. And this is nicely captured in this beautiful quote, I think, from Chuck O'Brien. So in the Lancet, at some point after continued repetition of voluntary drug taking, the drug user loses the voluntary ability to control its use. At that point, the drug misuser becomes drug addicted, and there is a compulsive, often overwhelming involuntary aspect to continuing drug use and to relapse after a period of abstinence. This is what we're talking about, compulsive behaviour. And you can study this in rats, and the way we studied it is with self-administration, but the added nuance of having electric shock delivered if the rat continues to self-administer. And it so happens that our high impulsive rats don't care about electric shock, and they continue to self-administer the drug. Although they're just as sensitive to pain as normal, they're not more sensitive. These animals persist in this behaviour, despite the adverse consequences, which we think is a good operational definition of compulsive behaviour. And as you can see, resistance to punishment is predictive of this tendency, as is impulsivity. Hence, the hypothesis, Barry Everett and myself, from impulsivity mediated by the ventral stratum to compulsive behaviour. And here the idea is that, as you'll hear, an important element of compulsive behaviour is the tendency to form habits which get out of control of the frontal cortex. But as we know, again, from rodent work and also some human work, the dorsal stratum is more implicated in the control of habits. I'll be defining what I mean by this in a moment. And also, indeed, in terms of relapse susceptibility. So our concept of addiction is from impulsivity, if you will, from ventral striatal circuitries, where the drugs have their initial molecular site of impact to control by the dorsal stratum, with a probable wiring circuitry to go with that, involving these loops, which Haber has shown do communicate in this general vector from the shell region of the accumbens, where we think drugs have their initial reinforcing impact, all the way through to the caudate and the putamen via these cascading spiral loops, which connect the shell ultimately to the putamen. Plus this progressive loss of top-down control from the frontal cortex, produced both constitutionally, it's there beforehand, as you've seen, but also potentially because of the toxic effects of the drugs themselves. A double whammy, which impairs control and drives you towards autonomous behaviour mediated by the dorsal stratum, as well as the ventral stratum. So some of the evidence for this, and I can't show you all of it in this lecture, is this study by Barry van der Schuren, where they infused dopamine receptor blockers into the dorsal stratum while rats are performing this self-administration behaviour. And you can see, interestingly, when infused into the dorsal stratum, after a good deal of training, there's a beautiful dose-dependent reduction in responding, which is not simply a motor effect. Whereas in the nucleus accumbens, at this stage, the drug loses its efficacy. Although the behaviour is initially controlled by the nucleus accumbens, and you need the nucleus accumbens to acquire this behaviour, with training it becomes less important, perhaps, than the dorsal stratum. That's the idea. Now let's switch to another form of compulsive behaviour and compare it directly with stimulant abuse. So this is obsessive compulsive disorder. And this is a study with Ed Bormore and Karolinska and others using the so-called resting state functional imaging paradigm, where the subject lies flat in the scanner with their eyes open, not thinking about anything in particular. And you measure the fluctuations in the bold response among 50 regions of interest, in this case just in the cortex. And you look at the intercorrelation of activity among these 50 sites as a proxy measure of functional connectivity at that point. It's, quite controversial methodology and needs to be further validated, but interestingly in this study, it came up with the following answer for two groups of patients, substance-dependent, stimulant-dependent individuals and OCD patients compared to controls. And what we found is that two areas in particular were disconnected relative to the rest of the cortex in these patient groups. They were both the same areas in both of the patient groups. They're both in the frontal lobe. One is ventral medial and the other is frontal pole, superior orbitofrontal. And the interesting additional result is that the degree of connectivity here was inversely related in both cases to a measure of compulsive drug use in the stimulant drug abusers, the Opta scale, and the Y-box measure of OCD in the OCD patients. So the functions are quite parallel for the same brain area. They're both measuring compulsive behaviour, and they're both in the same area, suggesting there is something in common here, a construct which we might call compulsivity. If you actually look at OCD symptoms in stimulant abusers, we do see them. So this is the study of Karolinska. The impulsivity data I showed you earlier are here, with the addition of another group of cocaine abusers who are recreational users, who we find in the financial houses, often in the financial centres in London, who are quite wealthy individuals. They abuse a lot of stimulant drugs, particularly at the weekend, but they haven't yet acquired a diagnosis of compulsive substance abuse. And as you can see, they don't show this phenotype. So mere drug exposure probably isn't enough. This may relate to the high degree of individual differences that we find in subjects proceeding to stimulant drug addiction. And you'll note that sensation seeking doesn't act as a phenotype except in the recreationals it's extremely high as stimulant-dependent individuals. So we see a difference between impulsivity and sensation-seeking. But a major interest for this talk is the scores on a measure of OCD-like behaviours. And again, you can see this same pattern. The siblings are showing even OCD behaviours more than controls, and stimulant-dependent individuals are showing a lot of OCD behaviours. Check-in, disgust, contamination worries, and so forth. So what is compulsivity? I've already defined it and identified prototypical disorders. But what does it mean at a psychological level? I tend to think of this in terms of a hierarchy, and at the bottom of the hierarchy are simple motor acts, which can become stereotyped, almost like tics, and are seen following chronic amphetamine abuse, as punding, for example. We've seen the impaired ability to stop a response or sequence, but also a persistence, I'm going to argue, in habits, or in situations where you have to show some change in behaviour, when rewards are stopped, as in extinction, or in reversal learning, where the contingencies change. And at a more conceptual level, inflexible thinking or strategies. All of these contribute to compulsive behaviour, this focus on a single goal, perhaps, or even beyond a single goal. And this derives from the learning theory of Tony Dickinson and others, that for instrumental or voluntary behaviour, we have two main categories. That behaviour where a response or action leads to an outcome or reinforcer, for example, a drug or food. This is voluntary behaviour, quite normal. This may occur in drug use early on, where you're trying to obtain a given effect of the drug as a consequence of your drug-seeking behaviours. But then, especially with repetition, we argue that the drug itself becomes perhaps a little less important, and the behaviour becomes more automatic or automatised, so that a stimulus directly elicits a response without reference to the goal. And so this may contribute to relapse, automatic drug seeking, which gives the behaviour a compulsive attribute. Now, to diagnose whether you have voluntary or habit-based behaviours, one technique is to devalue the goal and see if the behaviour continues. If it does continue, then presumably it's habit-based. This is a major hypothesis. And using that hypothesis, other investigators, including O'Doherty and Dickinson, have shown that with fMRI in humans, as predicted from experiments with animals, the goal-directed system does involve the ventral medial prefrontal cortex, here in the eyes, as well as the caudate nucleus, whereas the habit-based system implicates the putamen and probably motor circuits. Now, just taking snapshots structurally in the brain in stimulant drug abusers gives us a handle on these neural substrates of compulsion. So, for example, here we have stimulant-dependent individuals versus controls. These are horizontal sections. The blue signs are reduced grey matter from structural MR, and the red and yellow are heightened grey matter. And you can see, interestingly, a huge reduction in frontal areas in grey matter, particularly in the ventral medial prefrontal area, which is actually a function of the duration of stimulant use. So there's probably a toxic effect going on here, which may not be permanent, but certainly reduces grey matter the longer the duration. And intriguingly also, we find bigger basal ganglia in these subjects, as you see here, caudate and putamen. By contrast with the recreational cocaine users, these often have thicker frontal lobes. So this may be a resilience factor to prevent these individuals from becoming addicted. They hold their jobs down, they're very smart, they're taking a lot of drugs, but they have thicker frontal cortices. The unaffected siblings, remember the endophenotype study, even they have some evidence of increased putamen and caudate and amygdala size and grey matter, suggesting a propensity to use the habit-based system. How do you get from habits to compulsions, though? This is tricky. I mean, habits, are quite simple things, and we all have habits, but how do they become compulsions? We don't know. But here's some suggestions. One is sensitization, as proposed by Robinson and Berridge. We know that exposure to stimulant drugs does seem to bias the system towards a habit-based system. That's from experiments in animals largely, but also some evidence in humans. Stress is another factor, which we know biases the system away from goal-directed behaviour to habits. And that's certainly evidence for that also, and we know that individuals who become drug addicted are under stress, so this is clearly a major factor. But we also think a major factor is this impaired top-down control, which may even pre-exist drug exposure. So if you don't have top-down control, your habits get out of control and may develop into compulsions. That's the hypothesis. Well, to test some of these ideas, we've been focusing on OCD. You may have obsessions about car crashes, which produce anxiety, which lead to compulsion. So this is the traditional model of OCD. We argue that compulsions are very important to study in OCD. This has been our focus rather than obsessions. They're easier to measure, and we think they derive in part from increased habits which are out of control. So this kind of linear causal account of OCD, that you're anxious, you have obsessions, and you relieve the obsessions by producing these behaviours which are compulsive, probably has some truth to it, but we find some problems with this account. So these problems include the fact that compulsions can generate anxiety as well as relieve it. They can include covert mental acts, so compulsions can be mental. With these parallel cortico-striatal loops, they're not only controlling actions, they're controlling cognitive outputs as well, mental acts, such as mental repetition, counting, mental reviewing, checking, and so forth. Compulsions can occur in the absence of obsessions. And the evidence for pure obsessions in OCD is very debatable. So, whereas compulsions always occur, obsessions are less obvious. Consider compulsive hoarding, which often occurs without very high Y-box scores. We think this is an example of OCD, in fact, without obsessive behaviour and without anxiety. Cognitive behavioural therapy based on anxiety relief is not especially effective in OCD, and nor are anti-anxiety agents, such as the benzodiazepines. What you need for OCD is response prevention and exposure, so you have to work on the behaviour rather than the cognitions in OCD. And I think perhaps recognising this, DSM-5 is beginning to separate OCD from the anxiety disorders. It's now in its own spectrum. So that's the background to some of these studies, which we've performed on a large number of OCD patients. This is a typical sample, and of course they're anxious. They're often depressed, but I should say that we deliberately try to choose subjects with low depression scores to at least reduce that possible contamination of that comorbidity. And what we've been doing is using different tests of habit learning, goal-directed versus habit learning, both in OCD patients and also in stimulant drug abusers, to test this idea that there's a drift, a bias towards forming habits. So we have three major paradigms here. One, a computational paradigm, which Valerie Voon and I have used, has now been published, in fact. An avoidance habit paradigm and an appetitive behaviour paradigm. Let's have a quick look at these. So, first of all, the computational paradigm is very complicated. It will take me probably half an hour to describe it all, so I'm just going to simplify it in a couple of sentences. This is a complex decision-making task where the subject really, in order to be successful, has to form a model of the task, so we call it a model-based task. What that really means is understanding all the contingencies and intricacies of the task, having a big picture of what's going on. And the big picture of what's going on may mean that you have to make some choices which seem disadvantageous at the time, but will be advantageous in the future. Okay? Versus so-called model-free learning, which is directly responding to reward or punishment contingencies. So this happens, you were punished or rewarded, and your behaviour is entirely dependent on that. And we humans use both of these strategies to do this task, and you can separate them out computationally in terms of the patterns of behaviour that you produce. So the only other thing you need to know about this is you can measure the proportion of model-based, that's big picture strategies, against habit strategies with a single parameter called omega, or W, as I'm going to call it. And that bias score is shown here, and this is a validation by my student Claire Gillan of this task given to a very large number of subjects, healthy volunteers, on the web, as well as a huge number of questionnaires, eating disorders, OCD, schizotypy, depression, anxiety, and so forth. And all the questions are represented here. What she did was a factor analysis of the questionnaires and derived 3 factors. One she calls anxiety, another she calls social withdrawal, and a third one which looks a lot like compulsivity. And the compulsivity factor correlates quite highly with the bias produced to habits in this task. So this is the validation of this procedure. Valerie Voon and I and others have used this same parameter, this proportion of habit-based to goal-directed behaviours. The lower the score, the more habitual, the higher the more model-based, in a range of different pathologies as well as healthy volunteers. These are binge eating disorders, obese subjects, OCD and stimulant abusers. And you can see in 3 of these groups, not the obese, this drift towards habit-based processing. So there is this propensity, even with this abstract task, to use habit-based strategies in these groups. Okay. So another way of measuring them is to have a game, the fabulous fruit game, where you design these contingencies where a stimulus, let's say a fruit, may lead to a rewarding fruit if you can remember the right action, left or right button, associated with these fruits. You get points, and OCD patients are perfectly okay at learning this task. But then what we're going to do is devalue some of the fruits to see whether your behaviour is habitual or goal-directed. So you can devalue the fruits, these fruits in the boxes, as you see here. And when you do that, you can then give 2 tests of goal-directed and habitual behaviour. The first is a slips of action test. And what you do here is you present the stimulus fruits as quickly as possible on the screen, and you either have to respond or not. And of course, what you should not do is respond when you're responding for a devalued fruit, which loses you points. But as you can see, the OCD patients do over-respond for these devalued fruits compared to controls. And their tendency to do this correlates quite highly with their Y-box OCD symptom score, with no correlation with anxiety or depression scores. So this is again a lab assay which captures some of the habitual automatic tendencies to respond regardless of the value of the fruit in this case. And if you actually quiz them about their knowledge for outcomes, it's very poor in OCD patients. So you can quiz them either behaviourally or with a questionnaire by basically asking them, do you do the left or the right response in order to get this fruit? And they're very poor at that. So again, the bias is towards habitual behaviour away from goal-directed action outcome knowledge. We've used a similar test for stimulant abusers. This is Karolinska. In this case, we used animals rather than fruits because for some reason, drug abusers are very keen on animals. They like this test, but they don't like the fruits task. Interesting, exactly the same test, but with animals. And we see much the same kind of thing in our stimulant abusers. They're actually a bit worse at learning but they get there in the end. However, with the actually quizzing them about what you have to do in order to earn your animals, they're rather poor, both by questionnaire and by behaviour. And when you do the reverse test, you do the test where they have to control their actions in order to win the valued animals as distinct from the unvalued animals. They actually make more responses. So they show this automatic tendency to respond, which isn't a general tendency, because if you devalue stimuli rather than outcomes, although they're impaired a bit, they're not impaired. This executive working memory problem is not sufficient to account for their problem in controlling habits. So we see something in parallel between OCD patients and stimulant drug abusers. The third task is an avoidance task, an active avoidance task. This is a pretty simple test. You're equipped with 2 electrodes on your wrist, which are going to give you mildly painful shocks. And you can avoid these shocks, which are signalled by stimuli, blue for the right, yellow for the left, and red is safe. You can avoid these shocks when you see these stimuli if you press the appropriate foot pedal, left for left wrist shock and right for right wrist shock. So subjects learn this very quickly, within about 9 trials. But then we can devalue the shocks, and the way we devalue them is very straightforward. We just simply disconnect one of the wrists, but not the other. So, and we carefully explain to the subject that we're taking the electrodes off here. You can't be shocked. Do you agree? Yes, no, I'm quite safe, it's fine. I'm okay there. But they will be shocked on the other wrist. The idea is to see whether, nevertheless, they will persist in this behaviour when they see the CS, for the devalued versus the valued shock. And what we see is quite a large deficit in terms of OCD patients over-responding in this phase for the devalued shock. We've replicated this. So this is a very simple situation, clearly. It doesn't continue indefinitely. They only over-respond 1 or 2 times, maybe, but they do over-respond. They have this tendency to keep going when they don't need to. And this is correlated subjectively with an urge to respond. When you ask them, why did you do that? Oh, I don't know really, I just felt I had to do it. Premonitory urge, very highly significant. Now we've imaged this, and not all OCD patients will show a huge effect on this task. About half of them do. And you can see, we can then compare these 2 subgroups of OCD patients to see in the brain where is the difference between the subjects who are showing the habit-based persistence and the ones who aren't. And the answer is in the caudate nucleus, actually, which is quite where we predicted, but it's certainly in the basal ganglia. So the group who are tending to show this premonitory urge and this habit-based persistence have overactivation of the caudate nucleus. Additionally, when you measure the so-called psychophysiological interactions with other areas, we find that they have a reduction of activity in the inferior frontal gyrus. That's exactly that area which we've identified to be involved in behavioural inhibition. So this area is less effectively controlling the caudate nucleus. And we also find an increased activity in the subgenual region, which in the rat brain we know is the toggle switch, which can toggle the animals between goal-directed and habitual strategies. And when it's more active, it promotes habit learning and in rodents, and this is what's occurring in our OCD patients who are showing this habitual-based behaviour. So there's an interaction here between frontal mechanisms, top-down mechanisms, and basal ganglia structures in mediating this persistent behaviour. Okay, well, I'm going to continue by asking a couple of other questions about compulsivity. And finally, I'll talk a bit about anxiety in OCD, and then we'll finish, okay? Should be on time. So the thing I'm going to focus on for compulsivity, finally, is rigid thinking or strategy. And this is customarily measured in human neuropsychology with the Wisconsin-Cardinal test, which I'm sure you all know is a test which is quite sensitive to frontal lobe damage, as discovered by Brenda Milner. So simply speaking, it's a rule learning task. You have to learn what is the correct principle for sorting these cards. Is it colour, shape or number? Once you've learned the rule may change. You're not told that. The clinical psychologist gives you feedback, and if you persist, that's incorrect, of course, and it's often associated with frontal lobe damage. Well, our monkeys in Cambridge don't like this task. They can't hold the cards, and they do unmentionable things to them, throw them up in the air and so forth. So you can't test monkeys with the Wisconsin-Cardinal test. So we use a touchscreen version of the task in the Cantor battery, where we have 2 dimensions rather than 3, shapes and lines, and the monkey has to work out which of these shapes is best correlated with banana juice, ignoring the lines. And then at some point, the lines become relevant, and that's equivalent to the Wisconsin-Cardinal shift, okay? If you understand. So they begin by learning a compound discrimination. A compound is when you have 2 dimensions. We may give them examples of new shapes and lines to make sure they've understood the rule that they should attend to shapes, for example. But then, when we give them new examples, at some point we may shift them so that they suddenly have to attend to lines. And that's the extra-dimensional shift, which is endemic to the Wisconsin. We can also test flexibility in another way, in an emotional way, if you like, an affective shift, by switching, keeping the stimuli the same, but switching which is reward and which is punished. This is called reversal learning. So this has always been rewarded, and this has always been punished, but now we're going to change it, so it's the other way around. That gives you a difficult decision. Should I go with something which used to be rewarded, but it's now punished, or vice versa? And you've got to show flexibility in this situation. And in our original study 20 years ago, Angela Roberts, Rebecca Diaz and I showed that in the marmoset brain there are different circuits controlling these shifts. So the reversal shift is controlled by the orbitofrontal cortex. So when damaged, these monkeys take much longer to reverse compared to normal monkeys, and also compared to control intradimensional shifts here. By contrast, their ability to use a conceptual shift is unimpaired compared to normal. By contrast, lateral lesions produce the opposite pattern. Here you get a deficit in conceptual, extra-dimensional shifting, but no problems with the simple reversal shifting. So 2 different circuits involved in cognitive flexibility. They're both implicated in OCD. Neurochemically, they're separate as well, because we've found quite a lot of work, which I don't have time to review in detail, that this extra-dimensional conceptual shift in the lateral frontal cortex is controlled by catecholamine mechanisms, dopamine and noradrenaline, whereas, intriguingly, serotonin is very important for the affective reversal. So there's a lot of serotonin transporter in the ventral frontal cortex. These are serotonin transporters. And we've used a lot of neurochemical methods for depleting serotonin in the monkey to show selective problems in reversal rather than extra-dimensional shifting. This is relevant to OCD because, as you know, patients with OCD are often treated with rather high doses of SSRIs, selective serotonin reuptake inhibitors, which promote increased serotonin. Nobody knows why. We don't think it's simply treating anxiety or depression. It's treating something else to do with compulsive behaviour. We've recently got further evidence on this from rat studies, where we've studied reversal learning in a very simple paradigm, where the rat first of all learns to go right, and then they have to shift to go left. And there's a lot of individual differences here in rats, just as there are in impulsive behaviours. And those rats that persist inappropriately, perseverate, they're the ones who have reduced serotonin in the orbitofrontal cortex. These are normal rats, but they are rather persistent, and they have highly significant reductions of serotonin particularly in the lateral orbitofrontal cortex. Also changes in 5-HT2A receptors, and also changes in the serotonin metabolite. So there is something to do with a compulsive trait here, which is serotonin dependent, and is remediated by citalopram and not atomoxetine, the noradrenaline reuptake blocker. Now we know this atomoxetine is very good at treating impulsive behaviour. That's what I spoke about in 2012 here. But now we know that citalopram is a much better treatment for compulsive behaviour. So that's one of our messages. Okay, well, as far as OCD goes, we do have a paradigm for measuring these shifts in humans. This is Adrian Owen's work. He uses faces and houses in this task, but it's the same principle as I've described. And in the bold responses, the ED shift is associated with increased ventrolateral prefrontal activity here, whereas reversal learning is associated with increases in orbitofrontal activity here, laterally in particular. And also in the parietal lobe, this network is activated. So we see in the human something similar to what we predict from the marmoset. There's two different circuits here. And we've used this paradigm in patients with OCD and their relatives who don't have OCD. And both of the relatives and the patients underactivate the orbitofrontal circuit, which mediates reversal learning, suggesting they have a problem in that circuitry. They also have a problem in the other circuitry associated with higher order cognitive flexibility. So this is the Cantab test as used in humans, and it runs very quickly in about 15 minutes. Subjects are initially trained to learn about shapes, and then suddenly they're shifted to lines. It's the same principle as the monkey, with the same stimuli, actually. And patients with OCD are very bad at shifting. As you see here, they take more trials to learn to criterion, as do their relatives. So these are the unaffected first-degree relatives. The normal learning is okay, but the shift is selectively impaired in these two groups. So cognitive rigidity may be an endophenotype in part for OCD. And in terms of medication, as we predicted from the monkey work, those subjects medicated on SSRIs don't show any difference in this ED shift deficit. So that brings that together again with the monkey work. Recently, Mathilde Vagui in our lab has used resting state functional MR to look at the connectivity between the ventrolateral, again, lateral frontal cortex 1047, and the caudate nucleus. And this functional connectivity is reduced as a function of impairment on the ED shift task in a large group of OCD patients. So we think we've identified the circuit in the human brain which is less active in these patients, controlling their rigidity, as again predicted from the marmoset work. Whereas on other cognitive tests there is no relationship, so this is not a general effect, it's related to cognitive flexibility. And we find this is relevant to treating patients with OCD. We've got a recent collaborative study with Eileen Joyce at the Institute of Neurology and the neurosurgeons there, Dr. Hariz, where we've been studying treatment-resistant OCD patients. Now, these are very severe patients who are spending most of their time in compulsive rituals and obsessions. So that no treatments have worked. They're completely treatment-resistant. And only very few subjects, of course, can be admitted to such a study. But in this study, we compared two sites for deep brain stimulation. The ventral capsule, ventral stratum, including the nucleus accumbens, and the subthalamic nucleus. So these individuals had 4 electrodes, 2 each on these two sites, to compare their relative efficacy. And I can show you the preliminary results. Both sites worked. So they initially had a counterbalanced series of STN and ventral capsule stimulations. This is the baseline score in OCD and depression scores. And then after that period they were combined optimally, and they had a late period of CBT, cognitive behavioural therapy, added as well. And you can see the overall effect is tremendous. I mean, these subjects, some of them are almost in remission from being extremely high OCD scorers. But both of these treatments worked, we think, for different reasons. The anterior capsule ventral striatal seemed to have a bigger effect on mood as compared to subthalamic stimulation, which is only barely significant. But on the shifting task, we found that it was a subthalamic stimulation which produced the greatest improvement, so errors were reduced. So these two different circuits, and we've confirmed that they do involve different circuits using tractography, seem to contribute differentially to the OCD syndrome, one affecting mood-related factors and the other affecting this cognitive rigidity. Quite interesting. Well, in the last few minutes, if I may, I just want to quickly refer to another study, and I'm just going to give you a snapshot of this, talk about the other factor, which is anxiety and depression, which undoubtedly plays a role in OCD, perhaps to promote this imbalance in habit and goal-directed behaviours, and in combination with failures in top-down control, leads to OCD symptoms. Okay, so in this paradigm we used a threat reversal paradigm where two faces are presented in the scanner. One is paired with shock occasionally, and the other one is safe. And subjects learn which of these it is, and you have to measure, this is Pavlovian conditioning, so there's no response they can do. But you can measure their galvanic skin response, the sweating, as an index of Pavlovian conditioning. And what we find in this task is that OCD patients learn this fairly normally. So this is the GSR response to the CS+, the shock-paired face, and the safe face. OCD patients are here. They show a similar differential. Maybe in late acquisition they get a bit impaired. But the interesting thing is, despite this impairment, when you reverse them so that the previously safe face becomes threatening and the previously threatening face becomes safe, healthy subjects learn this pretty quickly, as you can see here. This is the reversal. But OCD patients are horribly impaired on the reversal. And it's not because they have impaired increased anxiety. Their amygdala response is normal in these groups, pretty much. But what we find is that they have relative overactivity in this ventral medial prefrontal area, which is associated not with learning about the CS+, but learning about the CS-. So when you do the contrast between these two in the fMRI, it's negative in control subjects, positive in the OCD patients. And you can see over the stages of the task, it's pretty inflexible. It doesn't change, whereas the control subjects change in association with the change contingencies. So there's a kind of rigidity here associated with the failure to discern the safety signal. And actually, this hyperactivity predicts the impairment in reversal learning in the second phase. So before they actually get to reversal, you can predict the ones which will show the worst effects. And the other interesting aspect of this is that some of the OCD patients think they're being shocked by both stimuli in the second phase. And it's these subjects in particular which have the highest levels of ventral medial prefrontal activity. And this isn't simply just in fMRI, we also see it in resting state, so it's real hyperactivity of this area. And that leads us to a hypothesis that in OCD, there's an imbalance between these circuits, such that the lateral circuits tend to be underactive. The medial frontal circuits, including to the nucleus accumbens, tend to be overactive. And this may be a model to explain the drift from adaptive goal-directed behaviour to their compulsive habitual behaviours. Okay, so in summary then, these are different flavours of OCD: hoarding, trichotillomania, check-in, hand-washing. We argue that importantly, there's an imbalance in habit and goal-directed systems, a failure in top-down control as seen in the cognitive rigidity tasks, and this hyperactive medial prefrontal area which seems to be mediating some of the anxiety associated with this condition. So my collaborators include Barry Everett for the work on drug addiction, and also Karolinska for the human stimulant drug addiction work, and in particular Barbara Sahakian, but also Anika Perkis-Shute and Claire Gillen for the work on OCD. Now, clinical collaborators also on the right-hand side. Thank you very much.